Okay, now, aside from DNA, there is RNA. And RNA has all kinds of cool functions, and every time we turn around, we find more new types of RNA with new functions. Okay, RNA biology is hot as a firecracker field. There are all kinds of RNAs that play numerous roles in cells. Many of these RNAs, we still don't know what they do yet, but they're there. When I was taking cell biology, now granted that was a long, long time ago. That was 1976, I believe, maybe 77. That was a long, long time ago. Okay, you had only 3 major classes of RNAs, sort of the classical RNAs, and this is what they did. So I'm going to mention first our old classical RNAs. There is, of course, what we call the messenger RNAs, or often lowercase m RNAs. Okay, now what these are, of course, is they are an RNA copy, or actually more properly a negative or complementary copy, of a stretch of DNA, of a protein, the sequence of a protein coding gene. So messenger RNAs are actually copies of protein coding genes. Okay, and these are synthesized in the nucleus. You pull apart a strand of DNA, pull apart the strands of DNA in a local region around the gene, and the enzymes, RNA polymerase enzymes, read one strand of DNA and synthesize a complementary or opposite copy of RNA, just like we demonstrated in class when we talked about hydrogen bonds. Now that RNA, after appropriate processing, becomes a messenger RNA. It's transported out of the nucleus into the cytoplasm, and the messenger RNAs are what are actually read by the ribosomes, and the ribosomes read the instructions and assemble proteins together. More on that when we talk about ribosomes. Okay? So we'll say they're read by the ribosomes. So the messenger RNAs have the instructions that the cell uses, that the ribosomes use to synthesize a given protein. Okay, so that's one class. Another one, the transfer or tRNAs. tRNAs, of course, are also involved in protein synthesis. What they do is they have, each tRNA has a specific amino acid covalently bound to it. There are enzymes that do that. They take amino acids, they bind them to they do the condensation reaction to bind them to the appropriate tRNA, and then the tRNAs bind to certain parts of ribosomes, and effectively what they do is they carry amino acids to the ribosome, put them in the right place so the ribosome can join it to the growing protein chain. So we'll say carry amino acids and position them on ribosomes. So, not surprisingly, tRNAs are also involved in protein synthesis. Question back there.
Yes, what is the second arrow? What's below it?
The second arrow. Oh, this? Okay, read by the ribosomes. In other words, the ribosomes bind the messenger RNAs and effectively read the base sequence on that messenger RNA and join amino acids together in the appropriate order. The tRNAs also play a critical role in that process because they bind to the messenger RNAs and carry specific amino acids. Okay, so far that's protein synthesis too. Now finally, the ribosomes themselves are mostly made of RNA. So we have ribosomal RNAs or rRNAs. The ribosomes, our protein synthesis factories themselves, are made mostly of these ribosomal RNAs. ribosomes are made of, in prokaryotes, 3 ribosomal RNAs, in eukaryotes, 4 ribosomal RNAs. And they're one copy of each different kind. Now, the ribosomes, most of the volume and mass of the ribosomes are actually these rRNAs, especially the 2 really, really big ones. There are many proteins that are parts of ribosomes too, and they tend to attach on the outside of the ribosome RNA, so they sort of form. You can picture ribosome as having a protein shell and an RNA core for the most part. And as we'll see, the RNAs actually play, especially one of them actually plays one of the most critical plays a role in one of the most critical functions of ribosomes. Okay, now, if this was 1976, back this any time before the mid-1980s, that would be about it for RNAs, our 3 major classical categories of RNA. So we'd say, oh, RNAs are handling genetic information involved in protein synthesis. But now we see a lot of other roles of various kinds of RNAs. Because we know of new classes of RNAs as well. And every time we turn around, it seems that there's additional new classes of RNAs found. Okay, one of the first things about that RNA is not just rRNAs, tRNAs, and messenger RNAs, that RNAs have other critical cellular roles, was a remarkable discovery that won its discovery trip to Stockholm for a Nobel Prize. Now, one thing before we go on to, I'm going to mention one thing. Turns out that RNAs are not synthesized from scratch. All RNAs are synthesized by the process of transcription. In other words, you read a sequence of DNA and make the appropriate RNA. So there are genes, and the protein coding genes are used to make messenger RNAs. There are genes for ribosomal RNAs. In fact, since cells need lots of ribosomes, we have about 300 copies of those genes in our genome. Multiple copies of them because we need lots of ribosomal RNAs. There are tRNA genes. So all of those things are encoded in DNA. That's true with all RNAs. We could say all RNAs are synthesized, got that, using a DNA template. In other words, there are genes for all RNAs, not just the messenger RNAs. Okay, so that's a very important thing. You don't make RNAs from scratch. You make RNAs based on the instructions in a section of DNA that acts as a gene for that RNA. So all these other types of RNAs we're going to be describing in a few minutes, there are genes for those RNAs as well, scattered throughout the genome. Okay, well that's an important kind of thing, but first discovery of oddball RNAs. There was this biologist named Tom Chech out in one of the University of California schools. And he was studying a particular type of RNA that was found in a eukaryotic protist. In fact, a ciliated protist similar to paramecium and stuff. And he knows something kind of interesting about it. He noticed that this RNA was made as a larger precursor molecule, and then parts of it got cut out and the other parts got glued together. That was called Rna splicing. And he wanted to see what is causing this. What is grabbing this RNA, cutting a piece of it out, and gluing the other 2 pieces together. So he's looking for protein enzymes and stuff. He's looking for enzymes. He can't find any. And finally, he realized when he completely purified these RNAs, of these precursor RNAs of all proteins, there was absolutely nothing like that around, he would see that that RNA would still end up getting cut and spliced together. The only conclusion was that that RNA was splicing itself. It was doing hydrolysis reactions to cut a portion of itself out and then doing condensation reactions to glue the halves together. It would be like if I took a knife, a sword, and I cut down here, and I cut down here, and I threw the midsection out, and I glued the rest of me together. I wouldn't want to do that. It's not a really good idea. But, and I survived. But if I did that, my own version of Harry Carey, I guess. Okay, well, at any rate, that RNA actually could splice itself. And the process of splicing was two kinds of chemical reactions, plus recognizing specific sequences of the RNA. So that splicing reaction required recognition of specific RNA sequence. In other words, where to do the cuts. It had to do hydrolysis reactions, which are going to cut that piece out. And then it had to do a condensation reaction to join the remaining pieces together. In other words, that RNA had to have two different types of catalytic activities. One of them catalyze hydrolysis, and the other to catalyze condensation. So what that meant is that RNA was acting as an enzyme, a biological catalyst. So, Church discovered the first RNA-based catalyst or enzyme. Prior to that, all enzymes were proteins. When I was taking cell biology, it was drilled into your head. All enzymes are proteins. Enzymes are proteins. Enzymes are proteins. And you never forgot it. Turns out they were actually wrong. He discovered the first catalytic RNA. And he coined up the term because it was an enzyme that was made of RNA. He called these ribozymes. That discovery won him a trip to Stockholm. It was the first catalytic RNA discovery. Once that was found out, people were looking for other RNAs with catalytic activity. And they found them. Now it turns out that the vast majority of catalysts, the vast majority of enzymes in cells are proteins, not RNAs. But there are some RNA catalysts, in other words, ribozymes, and some of them have very ancient and very critical roles. So let's take a look at a few examples of ribozymes. Well, that cell splicing RNA is something that's unique to certain types of ciliated protists and stuff. Well, it was enough to discover the principle and get this guy a nice trip to Stockholm. Okay, but since then, there's, for instance, an enzyme called Rnase H. And Rnase is an enzyme that attacks and hydrolyzes other RNAs. Now, there are many different kinds of RNA digesting enzymes in cells, but this one was unique. It not only consisted of a couple proteins, it also had a small RNA in it. Doing various kinds of experiments, people realized the RNA was actually doing the dirty work. The proteins were just there to hold the RNA in the proper three-dimensional structure to attack it. In other words, basically hold the RNA so that you had the part of the RNA form the active site. So, what this is the actual catalytic part was that RNA. The protein just held the RNA in the proper three-dimensional structure for the RNA to do its dirty work. Now the other RNases, by the way, are proteins, usually small proteins. But this one is a ribozyme. Okay, another thing. It turns out one of the most critical functions of life requires a catalytic RNA. And that function of life, all life produces proteins. They take amino acids, read genetic instructions in the form of messenger RNAs, and they produce proteins. In other words, that's what the ribosomes do. And every cell from the lowest archaea to us has ribosomes in it. Now, prokaryotic ribosomes are a little smaller and operate slightly differently than the ones in eukaryotes. As a matter of fact, that gets exploited by fungal toxins that we now call antibiotics. They selectively inhibit prokaryotic but not eukaryotic ribosomes. So things like streptomycin and those types of antibiotics inhibit bacterial protein synthesis and not so much ours. Yes? Question? Oh, thank you. Question. All right. Okay. Well, it turns out, and this was suspected for a long time, but it turns out what actually catalyzes the condensation reaction that joins one amino acid to the next, what we call peptide bond, that's the covalent bond from one amino acid to the next. It turns out that peptide bond formation in ribosomes is catalyzed not by the numerous ribosomal proteins, but by the largest of the 4 ribosomal RNAs. A little bit of history on that. So, in other words, essentially the ribosome is actually a ribozyme, at least in this major function of joining amino acids together. And that large RNA, they sometimes call this as an crude indication size of 23S ribosomal RNA. That's what you find in prokaryotes. Eukaryotes have a somewhat larger version. We call it 28S. It's the same molecule, just the eukaryote version is a little bit bigger and has some extra sequences on it. Okay, but the key thing is that is the molecule that actually joins amino acids together. It's an enzyme. And like all enzymes, it has an active site. Now, a little history behind this. It turns out people could do experiments where they would successively strip off proteins off of the ribosomes. And then they would test these ribosomes to see if they did what they were supposed to do. And one thing they noticed is you could strip off almost all the proteins off the ribosome, so it was little more than RNA and maybe a protein or two still sticking to it. And although a lot of the ribosomal functions didn't work anymore, what did still work was peptide bonds were still formed. These stripped ribosomes, these nearly naked ribosomes, could still join amino acids together. They weren't all that good at it, but they could still do that. So some people are thinking, which was heretical at that time, you get burned at the stake for saying that in a paper or a conference or something, but people were starting to think, some of these people were thinking, is it possible that peptide bond formation is not done by proteins at all? Maybe the RNA has something to do with it? Because they could strip almost, but not quite all the protein. That was always the sticking point. Well, you haven't gotten rid of every single protein, so maybe the proteins that are still there happen to be the ones that make the peptide bonds. Okay. Now, second thing people noticed that was getting suspicious. Turns out that large ribosomal RNA, which is about 2,000 or so nucleotides long, it's a very big molecule. It's quite variable from one organism to the next, except in 2 short regions, a few tens of nucleotides each, and separate by a reasonable distance. Those 2 short regions are absolutely invariant. In other words, they do not change in sequence at all in any organism they've ever looked at, all the way from archaeans to us. You have those 2 little sequences that are wholly and unchanging. When they had the tools of molecular biology, they could modify these ribosomal RNAs and see what would happen. They found if you change as much as a single nucleotide on either of those 2 sequences, the ribosome would be unable to join amino acids together. In other words, peptide bond formation would be completely stopped. And any organism they tested changed just one base to another in either of those 2 sequences, and that thing's toast. It turns out that those sequences form the active site of the peptide bond formation. What the final clincher was when they actually got the 3-dimensional structure of entire ribosomes, the position of every single protein, every single amino acid, every single atom on that huge complex. What they realized, they knew from other studies exactly where peptide bond formation occurred on the ribosome, a specific region on the ribosome. When they looked at that, all they found in that structure was RNA, specifically that large ribosomal RNA. There was not a protein within a nanometer and a half, which is a huge distance in terms of biochemistry. So in other words, there's no protein around the area that peptide bonds are being formed. So that was the clincher. If there's no proteins around in that critical site, and all you have is that big RNA, the RNA is the one that's doing that. So that's a very important ribozyme. And think of this. The very first life on this planet must have had to have been able to synthesize proteins. Now, everything we have on this planet now, every living organism on this planet now has ribosomes. Go way back when, and probably something as large and complex of a ribosome wasn't used. But the idea that these two limited sections of that ribosomal RNA can form a catalytic active site that will catalyze peptide bond formation suggests that way back before the fossil record starts, that there were these proto-ribosomes that may have just been a relatively small RNA that positioned those regions in the right place to serve as a peptide bond-forming active site. Now, we don't have anything like that in the fossil record. Nobody's ever been able to pull that off in a lab setting, but that's probably what happened at the dawn of life on this planet. So we look at this ribozyme, and this goes way back to the beginning. Okay, that's another big one. Now, still another. This one has some important clinical interest. And this may not be completely a ribozyme. It may be that the active site of this enzyme is both, part of it is protein, but part of it is RNA. Certainly the RNA involved in this does a lot of the catalytic activity, but it may not be able to do it all on itself. So now it's believed that's a hybrid active site between part of a protein and part of an RNA. And this is an enzyme that is known as telomerase. And telomerase has a lot of interest nowadays. It's an enzyme. And the enzyme has several small proteins and a single RNA in there. And the RNA at least contributes significantly to the catalytic activity. So this is at least a half ribozyme. Now, what is telomerase and what does it do? Well, telomerase rebuilds things called telomeres. Now, what is a telomere? Okay, well, what happens? If you look at a eukaryotic organism, you have, here's the DNA stretched out from a chromosome. And there are hundreds of genes on that chromosome and control regions and all kinds of other things. Now, at the very end of the chromosome, there are these sequences of DNA, and they do not have any known genes whatsoever. And these sequences are called telomeres. And all chromosomes have them. Now, when a cell divides and the DNA replicates, what happens, and this is the nature of DNA replication, when you copy a strand of DNA, the way the enzymes work is you cannot copy every single last nucleotide there. You have a start point, and the first few nucleotides, or the first several nucleotides, are not copied. So you don't start at the very beginning, you start a little bit into them. And likewise, at the reverse end. So when a cell replicates, you lose a little bit of the end of each chromosome. So if you had genes there, of course, you'd be starting, after a number of rounds of cell division, you'd be starting to cut into genes, and that would be a bad thing. You'd eventually end up with non-functional or missing genes entirely. So we don't put genes on the telomeres. We have these other sequences that are kind of acting as a sacrifice bunt in a baseball game. Now, cell divides again, and we lose a little more telomere. So each time the cell divides, you lose some telomeres. Some of the telomere sequences. Now, what happens is when the telomere sequences in a cell get to a certain minimal level, it activates a set of processes where we activate enzymes that destroy all the DNA in the cell and then activate other enzymes that basically cause the cell to commit suicide. So you take an ordinary cell from your body and you put it into an appropriate culture media and culture it. Those cells typically have about 80 rounds of division before they run out of telomere and they self-destruct. It's not all that much. Now, if you start out with a newborn baby, that baby's cells probably have 75 or 80 rounds of division. You guys probably have 65, 70, something like that. I probably got maybe 50, 40, something in that range. Your grandma may have only about 20 or so left. Your great-grandma, if she's still alive, may only have a handful of divisions left. And then, as a matter of fact, it's considered that when cells die after the telomeres get beyond a certain minimum limit, well, if you start having cells dying all over the place, what's going to happen? Your organs aren't going to work as well. Maybe your skin helps to shrivel up and stuff like that. It's quite possible that slow death of cells by losing too much of their telomeres is a contributor to the aging process. Now, you look at some organisms, though. In some cases, some organisms produce this enzyme telomerase. And what happens with telomerase is it comes in, we'll call it TM. Telomerase comes in after a cell divides and it rebuilds the telomere. It builds it back up to the original specifications. It repairs it. Now, cells like that have active telomerase can divide indefinitely. Now, of course, single-celled organisms are like that. They have active telomerase. So I could take those yeast cells you were culturing, I could take some of those yeast cells, put them in another culture and grow them up and keep on doing that and do that over and over and over again for years, decades, even centuries if I was going to live that long. I won't, don't worry about that. Okay. So some of these cells, single-celled organisms, they rebuild their telomeres every time they divide. But when you look at multicellular organisms, that's a different story. The vast majority of our cells do not have active telomerase. They've got the genes for the RNA and protein components, they just don't activate them. So most of our somatic cells divide and they reach a certain limit and then they die. Now, cells inside a living body generally don't divide very often. They'll divide to repair tissue, repair injuries, things like that. Like I gave myself a nice blister, and some of the cells in my underlying dermis in the past week have been dividing to fill the area up that I rubbed off when I was doing something over the last weekend. Okay, and then they will stop dividing pretty much when that wound is completely healed. Now, why do we do that in the first place? Well, one thing is that if a cell turns cancerous, having that built-in limit on rounds of division can keep some cancers in check, because they can only divide a certain number of times before they run out of telomere, and then they die. Problem solved. But some cells do have active telomerase. What ones have active telomerase? Okay, well, one of the kind are the stem cells. Stem cells are something that's, of course, a topic, a very hot type of topic, especially in ethical issues and stuff. And stem cells are medical interest because a stem cell can turn into a wide range. If you use embryonic stem cells, can turn into any type of cell that the organism needs. They're the precursors, the progenitors of all these different cell types. Now, adults have stem cells too. Those stem cells have limited developmental potential. The first ones that were actually purified and analyzed are the stem cells in the bone marrow that produce all the red and white blood cells. So they proliferate, they slowly divide, and then when they get the right signal, some will proliferate, some of them will turn into precursors of red blood cells, Some of them will turn into cells we call megakaryocytes, and those would end up turning into platelets. Others turn into lymphocytes, the types of some of your immune cells. Still others turn into neutrophils and macrophages. So there's a whole bunch of different types of cells they can turn into. There are also even neural stem cells that, to a limited extent, may be able to repair tissue in the central nervous system. A lot of interest in that. There are stem cells that can repair muscle and connective tissue and stuff like that. They're present in very, very tiny quantities in an adult organism. And it's been very difficult to isolate, but they do have potential. The question is with stem cell therapy is we generally don't know how to turn a stem cell into a specific type of tissue or cell. And the other thing is we don't always know how to tell them to stop. So some of the experiments in animals and in humans have caused some real problems. What we do in general is you take stem cells, the appropriate stem cells or embryonic stem cells, inject it into the tissue that you want repaired or fixed up or what have you, and hope that whatever chemical signals are in that tissue will tell them to turn into the appropriate tissue type. One of the big problems with stem cell therapy is knowing those cells knowing when to quit. Many times it will continue to proliferate and effectively generate cancer. So that's stem cell therapy is although there has been some experimental success in animals and sometimes in humans, it's still we're a long way from having that as routine treatment. But it does have a great deal of potential, just we don't know enough about it yet. Okay, now, so stem cells will divide throughout the organism's entire life. If you're limited to a limited number of divisions, in other words, you lose your telomeres, that wouldn't be a good thing. You'd lose your stem cells. So stem cells have active telomerase. There are some of the few cell types in your body that keep their telomerase active because they need to constantly divide over a period of time throughout the organism's entire life because they're the reservoir for new types of cells. Okay, now, that's the good part. But the dark side of the force. If you look at many of the nasty, aggressive, metastatic cancers, lo and behold, they too have active telomerase. Not all cancers, but generally the nastier kinds of cancers, the ones that have spread and proliferate all over the place. They have active telomerase. Now, case in point, an example of this. The first human cells that were successfully cultured were done by a physician at Johns Hopkins University who was treating this woman in Baltimore named Henrietta Lacks. This was back in like 1952 or 1953. He took some of that tumor tissue and then he tried to culture it, and he was successful in doing so. He called those cells, which are cancerous versions of epithelial cells lining the I think it was the uterus. I think it was uterine cancer that she had. He called those HeLa cells, short for Henrietta Lacks. Those cells, in a proper culture, will divide once every 24 to 48 hours. Now, how many days has it been since 1952 or so? Those cells are still grown in labs all over the world. In fact, the total mass of HeLa cells in research labs would probably equal many, many, many Henrietta Lacks, and she was not a small woman by any stretch of the imagination. So there are probably literally tons of her cells grown all over the planet in research laboratories. Those cells have undergone thousands and thousands and thousands of rounds of division, and they're still going strong. In fact, they are so aggressive that if a little tiny droplet containing them gets into another culture, they'll quickly take it over. Just like the original cells took over that woman's body and killed her a few months after those cells were extracted. So, aggressive cancer cells, telomerase, they can reproduce, they can divide without limit. In this case, for many decades, for over 50 years, in fact, for 60 years. That's a lot of division, dividing every day or two. So, some of the most aggressive cancer cells have active telomerase, meaning they pass that limit. They can divide as long as they want. They can basically divide until you die. Okay, bad thing. All right, now, question is, how do they get that way? Now, many cancers are actually very slow growing. In fact, the people do autopsies on people who died of something else, and many times they find tumors in various parts of the body, but those tumors are very slow growing, and they may never cause a problem. It's the ones that are fast growing and tend to spread to other tissues. That's the ones that require treatment. For instance, prostate cancers are often very slow growing cancer. Many times, rather than doing surgery or radiation or chemotherapy on people with prostate cancer, if it doesn't look like it's aggressive kind, the doctor will just say, hey, come back. We'll take a look at every 3 months, every 6 months, just make sure it's not getting out of hand. But chances are you're going to die of something else before this becomes a problem, so I wouldn't worry too much about it. And that's common nowadays for some of these slower growing cancers. Okay, well, what happens when we get these hyper-aggressive ones that can proliferate rapidly, proliferate without limit, have active telomerase? One possibility is some cells somehow turn on the genes for telomerase. Maybe they do that. But there's another hypothesis. There's a lot of controversy nowadays about this. Well, what cells already have telomerase? Stem cells. Is it possible that the worst of the worst cancers are actually cancers of the appropriate stem cells? That could be the case. There's a huge debate about that, whether the worst of the worst cancers are coming from stem cell precursors or coming from adult cell, differentiated cell precursors that have somehow gone really, really bad, including turning on their telomerase genes when they had them turned off for a long, long time previously. So there's a debate about that. So it's possible that many of our worst cancers are actually derived from stem cells. They already have active telomerase, and they partially differentiate, but not completely, and they just don't know when to stop, when to quit, when to stop dividing, and they end up going and colonizing all kinds of other parts of the body, leading to eventual sickness and death. So that's an interesting kind of thing. Needless to say, there's a lot of research on telomerase. Possibility of things like specific telomerase inhibitors may slow down the progression of certain kinds of cancer. Now, the downside is it would probably slow down the proliferation of ordinary stem cells as well. Other people are thinking that activating telomerase across the board, not in cancer patients though, may actually slow down the process of aging. Although I don't think they've done it yet, I think they're gearing up to try to see if they can do that in like mice or something like that, and see if they can actually prolong and slow down aging. So telomerase is an interesting thing, considering that it's at least a partial ribozyme. It's a very interesting example. Now what we're going to do on Monday, I guess there's a good place to call it.